What's happening, guys? Aruj again, and today we're talking about mitosis and cytokinesis. The last time we met, we gave an introduction to chromosomes, and from there we went into the cell cycle, which started off with interphase. Interphase was divided into three parts. We had G1, where the cell grew, S, where DNA was replicated, and then G2. Essentially, by the time the cell gets to this portion right here, G2, it is getting prepared to enter mitosis. Mitosis is division of the nucleus, so that is division of the nucleus, while cytokinesis is division of basically everything else, division of the cytoplasm. Okay? So before we get into these two processes, I want to show you this picture. And I want you guys to think, what is actually occurring between these different cells? These are actually onion cells. And you can see that each of these cells are at different stages of the cell cycle. And I just want you guys to think, what is actually occurring in each cell? So if you were to look at, let's say this cell right here, you could see that there's not very clear defined structures in the nucleus, not yet. And if you were to look at, let's say this cell over here, it's even lighter. So what's going on with these two cells in this nucleus? Well, in this one, the chromatin is starting to condense, whereas this one right here, it's still in a chromatin state. But here it's starting to condense, starting to get darker. And if we look at, let's say, this cell, it looks like these chromosomes are actually moving away from each other. If we look at this cell right here, we can see that the chromosomes are actually lining up in the middle. Okay, so what's going on is all these cells, they're actually going through cell division. Now, why do we have to understand cell division? Why is this concept even important? Why are there scientists that study this process? Well, for number one, this is how we literally go from a single-celled organism, because a sperm hits the egg and it makes the very first cell, the very first body cell of us, and it's only one cell big. But now as we grow older, we end up, as young adults for you guys have about 37.5 trillion cells. So how do you go from one cell to 37.5 trillion? That's because of mitosis. It's by cell division. The cell is constantly growing and splitting, growing and splitting, growing and splitting. Another reason why we have cell division is for replacing old or damaged cells. So if you get a boo-boo, if you get a cut or a scrape, how is your body going to repair that? Those damaged cells go through cell division, okay? So over here we have growth and development, okay? We're going to grow more cells. Over here we're going to replace old or damaged cells. And the last reason why people study cell division is because it's actually a form of a disease. It's actually what cancer is. So cancer is uncontrolled cell division. Okay? So in the article that we read talking about the cell cycle, we went over that there are certain checkpoints and there are certain regulations inside of the cell cycle that tells it when to stop growing, when it's time to replicate DNA, and when it's finally time to grow some more, then divide. Well, in a cancer cell, those regulations, they're all out of control. And basically this cell has its gas pedal on growth and division, growth and division. So you end up seeing, getting this mass of cells, which actually is a tumor. And that's what cancer is. It's uncontrolled cell growth, okay? So these cells could cause damage to the nearby tissue. They could get so big that they end up stealing all of your nutrients. So it'll starve these nearby cells, and it'll cause lots of problems. But that's what cancer is, uncontrolled cell growth. Some cells divide at different rates. So we have skin cells. These divide once every few days. So what happens on your skin, these cells at the basal layer, at the bottom layer, as they grow and divide, they're going to work their way upward until they reach the surface. Now, why do they have to do that? Well, these cells on top are constantly getting sloughed off. Okay, they're actually getting sloughed off your skin. so they have to be replaced frequently. Okay. Your intestinal cells, they go through cell division about once every 2 to 4 days, and for the same reason. Just as your skin cells, they come across a lot of friction, they get sloughed off, the same thing happens in your digestive tract. As you're eating food, that food is sloughing off cells in your GI tract, and they have to be replaced quite frequently. Okay. But now, what about other cells, such as nerve or muscle cells? Well, they almost never go through cell division. And in fact, that's why nerve damage is very, very hard for it to get better, because those cells very, very seldom go through cell division to replace older damaged cells. Skin cells, on the other hand, they constantly go through cell division all the time. Okay, so different cell types will divide at different rates. Some all the time, some almost never. Okay, and if we look at the cell cycle, let's see how all this fits in. We have interphase, which is the longest part. Then it goes through mitosis, which is division of the nucleus. Okay, and then we go through cytokinesis, which is division of basically everything else. G1, the cell grows. S, we're going to replicate some DNA. G2, we're going to grow some more. We're going to get an extra centrosome. We'll see why that's important in just a moment. We go through mitosis, and there's actually 4 stages of mitosis, and I want you to remember those as PMAT. Okay, stands for prophase, metaphase, anaphase, and telophase. For short, I want you guys to remember PMAT, and then finally we go through cytokinesis, okay, which is division of the cytoplasm. Here's what this looks like. We have our cell in interphase. Notice the DNA is in its chromatin state, okay, loose spaghetti in the nucleus. The cell is going to grow, goes through S phase, DNA is replicated, goes through G2, the cell is going to grow more. We have our second centrosome. Now let's go into mitosis. Let's go into division of the nucleus. So what's going to happen is this chromatin is going to consolidate into chromosomes. When that happens, this nucleus is actually going to start to disappear. It's going to break down. And the last thing that's going to happen is these 2 centrosomes are going to move to opposite ends of the cell. So here's what this looks like. Okay. The nucleus is disappeared. Chromosomes are now visible. It's no longer chromatin. Centrosomes are on opposite ends. Now, a little mnemonic that I help to remember this is the P in prophase. I think of pair up, because we're going to get those 2 sister chromatids, and they're going to find their match, and they're going to meet up with each other to form that X-like chromosome. Okay. Next, we go to metaphase. The M in metaphase, I think of meet in the middle. Okay, so all those chromosomes, they line up in the middle, and those centrosomes, they send out what's called spindle fibers. Those spindle fibers attach to the chromosomes. Okay, and remember, this is still considered 1 chromosome, but there's 2 copies attached. Okay. Next, we go to anaphase. The A, I remember as away or apart in anaphase, and the 2 sister chromatids are pulled apart. Okay, they're getting pulled to opposite ends of the cell, and the cell is actually going to continue to elongate. It's going to get larger or longer, okay? Next, we go to telophase. The T in telophase I think of as 2 nuclei, because the chromosomes have reached opposite ends. Once they get to opposite ends, they're going to decoil. They're going to go back into their chromatin state. Our nucleus is going to reform. And then, moving into cytokinesis, division of the cytoplasm, we're going to see the cell membrane start to pinch. Okay, it's going to start to pinch down here, and that's actually called a cleavage furrow. Cleavage furrow. And the membrane is going to continue to pinch down the center until that cell splits into 2 different cells. Let's get rid of that ink. There you go. And now we have cytokinesis. Again, the T in telophase I think of as 2 nuclei. Cytokinesis I think of as 2 cells. Okay, that's what we're left with. So if we go back to this image, can you identify each stage that the different cells are in? So I would say this one right here would be an interphase because I don't see those solid chromosome structures. Let's see. In this one right here, what stage would this be in? Well, I would say those chromosomes are lining up towards the middle, so they're somewhere in metaphase. If we go to this cell right here, what stage would this cell be in? Well, I could see that those chromosomes are getting split apart, so I could see that is anaphase. Okay, they're getting split apart. If I were to go to, let's say this one right here, notice how these cells are pretty small. They're two separate cells. They're very small. I would assume that these two cells probably just finished through cytokinesis. They just barely separated, and this is going back into the chromatin state. Okay, just a little food for thought for you guys. Let's go to the last slide. Let's do a quick review. Interphase, we had G1, S, and G2. That's where the cell spends 90% of its life. It's when it's going through its normal functions. Now we go to the stages of mitosis. Prophase stood for pair up. M was meet in the middle. A was away in anaphase. T stood for two nuclei. And cytokinesis was for two cells. Okay? These portions right here, that represents mitosis. All right? And that is the end for now, folks, and I will talk to you next time in class. See you later.